Hello, my name is Alain Moreau. I'm a full professor at the Department of Stomatology, Faculty of Dentistry, and at the Department of Biochemistry and Molecular Medicine at the Faculty of Medicine de l'Université de Montréal. I'm also the director of the interdisciplinary Canadian Collaborative Myalgic Encephalomyelitis ICanCME Research Network. Today I would like to talk about you and myalgic encephalomyelitis and share with you an update of our ongoing projects. On this second slide, here I'll represent the disclosure of the different research program and the source of funding that I'm receiving. Today, I would like to present a glimpse of what we are doing in my research center in Montreal about two main projects. One called MicroRNA, which stands for Multi-Omic Approaches to Resolve Myalgic Encephalomyelitis, and the second one called SCOPIMED for the study of COVID-19 survivor profile for detection of myalgic encephalomyelitis development. In both projects, I will cover the latest development that we got so far and to share with you some promising discoveries. First, I would like to explain our methodology, especially our longitudinal study of post-exertional malaise. As you know, post-exertional malaise is the cardinal symptom of myalgic encephalomyelitis. So in the context of MicroRNA, we are investigating each participant over a week, where previously the nurse have sent 3 health questionnaires, SF36, MEF20, and the SQ2, to allow to have a review of the health conditions during the last 6 months. At the first visit, the clinical nurse are going at the home of each participant because most of our ME patients are severely affected and they are housebound. So far, we have tested over 193 ME participants, as well as 70 sanitary healthy control. So during this first visit, the nurse is not only explaining the different phase of the test, but she's applying a neurocognitive testing called brain check, which will establish the baseline value of each participant. We will also request each participant to wear a biometric vest called ExoSkin, which allow to measure different physical parameters in real time, and each participant are invited to wear the vest at least 20 hours over 24, and it is mandatory during the night to wear the vest to establish or measure the sleep disturbances affecting each individual. We are collecting different biological fluid like the first morning urine during each day of the week, as well as collecting a saliva sample at the first visit, second visit immediately after the stress test, and at the third visit, plus 5 days following the application of the stress test to measure a different change in the saliva, including DNA methylation pattern. At the second visit, we apply what we call a stress test, which is a mechanical stimulation, as you can see on this slide, where we use an inflatable cuff that is applied on one arm and that apply a gentle therapeutic massage from 0 to 4 psi. This creates some kind of exorcist effect on each ME patients, and during 90 minutes they receive this treatment, and that create like the effect of a strong exercise on them that induce post-exertional malaise. At the same time, we use frontal electrode to measure by near-infrared spectroscopy change in the brain oximetry levels during the stress test. We are taking, at the end of the stress test, additional blood samples, as well as taking another brain check test to measure if the induction of post-exertional malaise induce changes in neurocognitive function. The following day is where the post-actional malaise is developing in each ME participant. And we know that this post-actional malaise is variable between individuals. That's why we are following them over a 5 days period. At the third visit again, we are still collecting morning urine, performing another brain check to see if the neurocognitive functions are the same, improve, or further decrease from one individual to another one. We also ask each participants to perform the DPMQ, which measure more specifically the development or the effect of the post-actional malaise induced by our approach. So to give you an example about the phenotyping, this is a real case, ME 183, where we monitor brain oximetry changes during the stress test. So as you can see on each graph, you see on the upper part is the relative change in the left, from the left electrode or right electrode, identified by R or L respectively, the level of total oxygen measured by the electrode applied on the front. As you may know or not, the oxygen level in the brain is between 70% to 61%, because it's a combination of arterial blood, which is rich in oxygen, and venous blood, which is poor in oxygen. So as you can see, we saw a kind of slight decrease. But if we look about the different components that are measured more specifically, so the bottom panels in the right and left side, you can see here that there are drastic changes. And on the right side, there is a brain blood flow increase, which is indicated by the increase of the purple slope, which is the total hemoglobin level, as well as by the red slope, which monitor the oxygenated hemoglobin, while the black line represents the desoxygenated hemoglobin. But on the left side, you can see that in the after 1/3 of the stress period, we have a dramatic decrease by almost 20% of the circulating hemoglobin and oxygenated hemoglobin, suggesting that a drastic and severe decrease in the brain blood flow. So we don't know yet the meaning and the mechanism behind this asymmetry, but this is something that we are capturing in some patients, and we saw some very different patterns that also help us to understand what is happening in terms of what is induced by the post-actional malaise that we are inducing in each participant. As an example of the longitudinal monitoring of neurocognitive function with the brain check test, you can see on this slide the different functions that are measured, which are ranked from 1 to 200. So if you reach 200, you are a genius. And of course, if you are ranking in the first percentile, you are very, very severely affected, because 99% of the population will have a better score than you. So that's why I use the avatar, the minion. So the purple is a very sick MECFS patient, and the yellow one, the regular minion, is the so-called genius or healthy person here. Going back to the patient that I just described you, the change in the brain oximetry, so the ME 183, you can see that there's a very decent score before the test for the different functions. So you have the global score at T0, which means baseline. And after that, you see the global score at T19, which means T90 minutes. So immediately after the application of the stress test, which declined, and the global score T plus 5 day, which means that 5 days later, when we ask the participant to do the test again, is increasing, which is something that we wish for most of the participants, although some of them are still developing their post-actional malaise. What you can see on this very specific example is a very severe decline, especially in the memory immediate recognition and memory delay recognition, which is often common in many MECFS participants. So we use this strategy to characterize in depth our participants, but where to start? So we decide to centered our study about the role of microRNA in myalgic encephalomyelitis. So allow me to go back to the basis first. As you all know, each cell contains the DNA, which is our personal database where everything is encoded. So this is where we know how to produce different protein, and we need this code in each our cells. This code is translated in RNA or ribonucleic acid, which is giving us, or at least to our cells, specific instructions to produce the protein, which are the final functional products that we need to be functional. MicroRNA are very small non-coding RNA, which act as what we call epigenetic regulator, and by attaching to the end of the messenger RNA will prevent the translation by the ribosomal machinery. So this is a very specific machinery inside of each cell to decode the instruction and produce the functional product called proteins. So one single microRNA can prevent or block, sometimes completely, the production of specific protein, which will lead to a disease. More importantly, one microRNA can regulate or target up to 200 different genes, and one gene can be targeted by more microRNA. So that's why these very small regulators are very powerful and are involved more and more now in molecular medicine and used for diagnostic or prognosis approaches for different diseases like cancer, like neurodegenerative disease, and myalgic encephalomyelitis is no exception. So in fact, last year we published in Scientific Report a first paper which has been downloaded more than 19 times since November 2020, which has been in the top 100 lists of Scientific Report. And this diagram, or this paper, we report the interconnectivity of 11 circulating microRNA with key features of myalgic encephalomyelitis like fatigue, mental fatigue, as well as a function of muscle, circadian rhythm, sleep disturbances, and also some key metabolic factors that are involved. Secondly, not only these microRNA are interconnecting with each other and targeting very specific elements that are impaired in myalgic encephalomyelitis, but also this strategy allowed us to stratify ME/CFS patient in 4 clusters that allow to predict or explain the severity of some symptoms, and more importantly, and we will cover that aspect in the few next slides, that it can predict the therapeutic response to different drugs or molecules that could be applied to treat myalgic encephalomyelitis. So for instance, one of these microRNA, the MIR155P, is involved in the norepinephrine transporter deficiency called NET, and leading also to postural orthostatic tachycardia or orthostatic intolerance. So on this slide, you can see in the diagram A and B, the expression level of this microRNA, which is more elevated in ME patient with a confirmed POTS or orthostatic intolerance diagnosis when compared to ME in black and LT control, symbolized here by HC. This is true at baseline, panel A, as well as immediately after the stimulation, the 90-minute stimulation, panel B. Interestingly, we can also, in panel C, using the self-reported questionnaire, the DSQ, at the question 51, where participant can report having lightheaded and different tachycardic symptoms. So we can see and confirm that our ME plus POTS or having orthostatic intolerance are indeed very different than the ones that having only ME without these symptoms. We also check for other questionnaires, and as you can see in panel D, for the reduced motivation using the MEF20 questionnaire, as well as panel E for the physical fatigue score, again with the MEF20 questionnaire. ME patient having POTS or at least orthostatic intolerance are sicker than the ME without these very debilitating symptoms. Interestingly, elevation of miR-155P, which leads to the norepinephrine transporter deficiency, improve also mental fatigue score. So on the diagram on panel A, you see the link as demonstrated by engineering pathway analysis that miR-155P is targeting SLC6A2, which encode the net transporter. On panel B, elevation of the expression seems to be associated with the better mental fatigue score, so improved mental fatigue, as compared to patients having a normal expression, high expression, or very high expression of this circulating microRNA. This is particularly interesting because the work of Arnold et al. Through a very well characterized clinical study, they observed the effect of duloxetine, a pharmacological inhibitor of SLC6A2 or NET protein, on ME patients. This group have shown an improvement also in mental fatigue using the same tool, MEF20 questionnaire, on patients taking the drug compared to patients treated with the placebo. So here, by two different approaches, one is using the duloxetine, which is a pharmacological inhibitor. The other one is the elevation of miR-155P, targeting the very same gene, with the same outcome, a reduction or inhibition of NET. Conclude the same thing on two different cohorts of ME patients. So this is a type of converging data that we need to reproduce more often in the field of ME/CFS to further assess the clinical validation of our results, but also their clinical utility. Another good example that we are close to predict the therapeutic response among different patient subgroups using our panel of 11 microRNA is with the very well-known molecule called Amplogene. Amplogene is known to interact, is an agonist of the toll-like receptor 3, which is also targeted by one of our macroRNA, the miR-6819-3P. So, in a very simple words, if you have a high level of this microRNA, the target TLR3 will disappear and will be very unlikely to interact with Amplogene. So, in other words, this is the case about 2/3 of our patients. TLR3 is disappearing, so there's no way that Amplogene can boost the innate immunity by interacting with the receptor because the receptor is gone. Same with Viagra, which has been also tested as an inhibitor of the phosphodiesterase 5A, PDE5A, as you can see here, and the very same macroRNA can exclude the target again. So this is, I think, it's very important, and I think we are on the eve of using precision medicine for ME/CFS patients, not only to predict severity of the symptom, but also, more importantly, to predict the therapeutic response. We recently challenged our panel of 11 microRNAs to distinguish ME from a fibromyalgia patient, or FM. On this slide, you see a comparison of ME patient versus patient having ME and fibromyalgia as a comorbidity versus group with fibromyalgia or LT control matches for the sex and age. As you can see, for each of these microRNA tested in these 11 panels, you see that systematically there's almost no expression, or at least a very low expression, of each of these microRNA at baseline, which allowed us to use this panel now to have a more molecular objective testing to confirm fibromyalgia testing versus ME diagnosis, as well as in some kind when patients report having both, we can now measure that with a very high accuracy. So these are real novelties that can translate work in the lab to the clinics and can be very useful to many clinicians to understand and validate the condition of their patients. Among other ongoing research projects, I would like to spend a few minutes on the role of thrombospondin or TSP1 in the pathophysiology of ME. Thrombospondin 1 is a matriciellular glycoprotein that playing many roles in different tissues and cells and is well known as a role as a vasoconstrictor, and it could be involved, and this is what we saw in the post-sectional malaise. So the rise in circulating thrombospondin 1 could lead to vasoconstriction of blood vessel, thus leading to a decrease in blood flow to the brain and could be, at least in part, responsible of creating the brain fog. In contrast, a significant or sudden decrease in circulating thrombospondin 1 could be associated with orthostatic intolerance, which may or not induce tachycardia as a compensatory mechanism. So too little or too high thrombospondin will create some damage in the body, especially if those changes occur following the induction of post-exertional malaise. A good example, we use thrombospondin as a marker to explore a possible mechanism underlying the cause of POTS or orthostatic intolerance. So in the panel A, we were able to measure in the blood of patients, so at baseline, very high level of thrombospondin, here over 25,000 nanogram per milliliters. And following the induction of post-exertional malaise using our stress test, 90 minutes later, this level declined by almost twofold with 10,819 nanogram per ml. This sudden decrease was associated in panel B by an increase in the heart rate. As you can see, we have a patient on beta blockers, which are very slow heart rate at around 50 beats per minute. And you see the spike, the increasing spike, that some of them reach well above changes of 30 beats per minute. So these real-time measurements being achieved using the ExoSkin biometric smart test and gave you a good example why it's so important to do this longitudinal monitoring in the time to capture the events linked to the post-exertional malaise. On panel C, we can also see the variation of thrombospondin or delta TSP1 as 90 minutes versus the value at baseline, and you see the most significant drop occur in the subset of ME patient with POTS or orthostatic intolerance as compared to ME without POTS or without orthostatic intolerance or LT control. Interestingly, panel D, we can further substratify a patient based on their cluster using our panel of 11 microRNAs. And again, we saw some enrichment in that negative delta TSP1 value in the cluster 2B, which is more profoundly affected by POTS, as opposed to the cluster 4, where there is a strong elevation of thrombospondin following the induction of PAM, which is more often affected by a brain fog. Also, change in the plasma thrombospondin level seems to be associated with memory impairment. So by doing longitudinal evaluation of the different neurocognitive function using the brain check test, we can also illustrate the change between the memory score as well as following a change at baseline immediately after the stress test or later on. So on the first panel, you can see that patient having lower rank of their memory impairment at baseline, so below the 15 percentile score. So at baseline, 80% were below 15 percentile, and the majority was above. Immediately after the stress test, we see that the number with less than 15 percentile increase and remain relatively important even 5 days later. And we can see that the individual having more memory impairments were the one that having a higher circulating level of thrombospondin that can be seen in these 3 graphs, more specifically baseline and also following 5 days later the induction of post-sexual malaise. So we are just at the beginnings to play around and try to correlate the different neurocognitive function changes with change in circulating biomarker. The elevation of plasma thrombospondin levels were previously reported to be associated with poorer episodic memory in patients with sickle cell disease. Another type of analysis is regarding the analysis of DNA methylation profiles. So we start to study identical twins that are discordant for MECFS. So one twin is affected, not the other twins. This is a very powerful approach because the identical genetic makeup, around 98% between the identical twins, make it possible to study the effect of the environment on the development of ME by studying, amongst other changes, in DNA methylation, particularly those associated with the disease. So, in other words, when a gene is hypo or unmethylated, as you can see with the green light post, the gene is well expressed or sometimes even overexpressed. But when the gene is hypermethylated, so where you see a lot of red dot, okay, it will be sometimes severely repressed, and you will see a significant decrease in the expression of that genes that will lead to less protein production. So using this technology, we study different couple of discordant twin, concordant twin, multiplex families having enriched in several affected individuals over several generations, and also unrelated MEKs and unrelated healthy controls. So amongst different targets, we identify a gene called SMPDL3B, and which was hypermethylated in the ME affected patient, as you can see here, with 0.62 versus 0.25 in the healthy twin. So we further investigate what this gene is doing in our body, and this is where it's coming, I think, very interesting. As you can see here, we assess the role, at least starting with the role that is known about SMPDL3B. So SMPDL3B is widely observed in macrophage and dendritic cells. So it's part of the innate immunity and is negatively regulate toll-like receptor at the surface of the cell membrane, as you can see on this diagram. Is also involved, and its role is consistent with a possible role during inflammatory processes. Equally interesting, SMPDL3B is involved in the conversion of sphingomyelin to ceramide, and this is quite interesting because several metabolomic or metabolic studies identify a drastic decrease in the conversion of sphingolipid in ceramide. So in 50% of men with ME, the ceramide were decreased, and up to 86% in women with ME, so suggesting that SMPDL3B is impaired, mutated, non-functional, or not enough produced. We recently reviewed a paper where there is a link between thrombospondin 1 and SMPDL3B, which you can see with the non-alcoholic fatty liver disease, which is a disease of the liver that affects people who drink little or no alcohol. And in the liver cell called hepatocyte, the elevation of thrombospondin repress the SMPDL3B expression. And the lower diagram here show you that if you do that on a cell in vitro and you just add recombinant purified thrombospondin protein, you see that you have an immediate effect and a rapid effect on the expression and production of SMPDL3B. So interestingly, SMPDL3B is linked to some feature about ME patient, and also it's linked somehow with what I showed you before about thrombospondin 1. So we decide to measure the circulating level of SMPDL3B, so in the plasma and urine sample of ME patient. And to make a story short, what we found is the fact that elevation of SMPDL3B, which means the circulating form or the soluble form, is associated with the increase of symptom severity. So as you can see on the upper panel with different questionnaire, so more soluble SMPDL3B is produced, so over 30 nanogram per ml, and more severe are the symptoms. And in the morning urine, okay, you can also measure the changes. And again, more you accumulate in urine, which means that there will be a less functional protein anchored at the level of cells, so the symptoms are more severe. So for us, this is a pretty interesting discovery because we can modify SMPDL3B level somehow. Recently, through a great collaboration with my colleagues in Norway, Dr. Harsting Flug, we get access to their sample and we blindly measure the level of SMPDL3B in the plasma of their cohort. And as you can see on the panel A, we saw a very nice correlation with symptom severity with the elevation of soluble SMPDL3B in the plasma of the ME patient in this cohort. Interestingly, and we saw the same thing in our cohort in Montreal, is we saw a decrease with the age, which makes some sense for different reasons. First, many studies reported that there's a kind of stabilization of symptoms over time, especially around 50 years. And this is what we see here with a steady decline with the age, suggesting that maybe estrogens underlie the cleavage or liberation of soluble SMPDL3B in the plasma or morning urine. We also saw differences between female and male. And again, we saw that in female, the elevation of soluble SMPDL3B is associated with the production of estrogen when compared to male or when compared to women after 50 years after menopause. So this is a very important finding to understand, and that can explain the differences between male and females. On this diagram, okay, I told you that there are some possible therapeutic options, and this is here, there's a big warning sign. So nobody should be allowed to test it on himself or herself, or even through asking their attending physicians to do it without the proper analysis and classification of the patient. But let's go back to my avatars. So the healthy minions, okay, have a lot of anchor SMPDL3B, which is the like eggs in the membrane, and few of them are freely liberated in the plasma. So normally in the healthy people, you don't have a tons of circulating SMPDL3B. In one subtype of patient with ME/CFS, you can see that there is more scissors. So there's an aberrant cleavage will release a lot more of solubles. So that's explained the high plasma level in some subset of ME patient. And we have also another subtype of patient that are very sick. Their problem is not the scissor, is not the enzyme that will release the SMPDL3B, but is the reduced capacity to produce enough SMPDL3B, which will end up with the same severity. And this is where changes, for instance, in DNA methylation could be involved in the low production of SMPDL3B. In both cases, therapeutic option will be very different. In one case, we need to prevent the access of the protease to the SMPDL3B protein. So one way to do that is to use a rituximase, because rituximase is known to interact with SMPDL3B. It's been already tested in clinical studies for different kidney disease. So it's not that efficient because it just simply creates an obstacle to reduce the access of the protease, but is doing some effect. But the most direct approach could be the use of GPase inhibitors that can pharmacologically target the protease and reduce their enzymatic activities. We're still looking for these specific GPase inhibitors, and developing the right platform, we can find them quickly. The other situation where we don't have enough SMPDL3B protein, we need to increase the transcription of that gene by different approaches like AMPK agonists, which can also involve the repositioning of drugs like metformin of N-acetylcysteine that can eventually are known to increase the transcription of AMPK, and also that will lead to more SMPDL3B. Again, these concept of therapeutic option request a randomized clinical trial for validation and a way to get more funding to achieve that. I would show you on this slide a short demonstration through my collaboration with Dr. Flug and Professor Mela in Norway. So we revisit the rituximab clinical trial, where we select only ME patients with elevated plasma SMPDL3B as an intent to treat at baseline. So this is what you see in panel A. So patients were stratified as a placebo at 0 before treatment and 18 months after treatment with the placebo, or at 0 with RTX, which stands for rituximab, and after 18 months with rituximab. So first, in the non-responder group, there's a big difference between the baseline and after treatment group, which we don't know why, but it's clear that there are a difference between both groups. In the B panel, where we look at the ones that respond to treatment, you can see that there is no significant change with the placebo group, which means that it must be something else, another mechanism. But at least in the rituximab group, we saw a dramatic decrease in circulating SMPDL3 as expected based on the previous role of rituximab to interact with that molecule. So again, this results retrospectively, where we reanalyze the rituximab clinical trial cohort, request a new clinical trial with the right questions, and to make sure that to whom rituximab could benefit. And also, this is a good example that rituximab may have additional off-targets effect that we must be aware of. The next few slides, I would like to talk about SCOPIMED, which stands for Study of COVID-19 Long Haulers Profiles for Detection of ME Development and possibly other type of long-term sequelae. So following a SARS-CoV-2 infection, you get COVID-19. So far, there are three options. You may have a quick remission or a slower one, but nevertheless a remission. Or you may end up with ME-like symptoms or fibromyalgia-like symptoms. And there are also other forms of long-term sequelae like neurological, respiratory, or cardio sequelae. So we try to understand the different trajectories and to define biomarkers that can explain the different trajectories and who or what is needed to do a long COVID versus why some individuals can quickly have a remission. We study primarily non-hospitalized asymptomatic or patient or the one having mild to moderate symptoms, and we select for the SCOPIMED, patient with at least a 6-month long COVID symptom. This is our minimum as inclusion criteria, but we have also we had access to other sample from non-hospitalized as well as hospitalized samples during either the acute phase or after the acute phase of the disease, through some collaboration with the Quebec Biobank, so BQC19, and the Canadian CanCME Biobank, where we have access to additional sample. The problem with these biobanks is the fact that all the samples are collected at different time point, well characterized using medical records, which is great, but they are not using our approach that use a provoke test to stimulate patient and see how they are responding to post-surgical malaise. This is exactly what we have done. So far, we are close to 50 patients with long COVID tested and 5 post-COVID patients that have a quick recovery. We use the same strategy that we used before with the Maestro ME protocol, where the same questionnaire before and the MPQ a week after. We are doing the brain check at baseline and immediately after the post-surgical malaise induction using the inflatable cuff. And we also are testing patient every 2 months, okay, which is called a visit 2, 4 months, visit 3, 6 months, visit 4, and visit 5, 8 months to measure when it's necessary sleep disturbances, okay, with the exoskin. Otherwise, we are simply collecting the morning urine, saliva sample, and of course, blood samples at different time. During the stress test, we are recording change in the brain oximetry level as well. So as a quick look using the self-reported health questionnaires, you can see here a comparison of SF36 and MAFE20 score between participants at first visit. So we can quickly see, so all the CAL means stand for long COVID and CO for post-COVID patient with rapid recovery. HC stand for healthy control and ME is ME patients. So you see that whatever the physical mental score with the SF36, we saw as by comparing group a lot of similarities. Same with the MEFE20 score. So the long COVIDs are mimicking a lot or looks like very much like MECFS patient. If we select few microRNAs, so we got some very interesting information. So we are using our panel of 11 microRNA to establish ME diagnostic, but some of them taking individually can also be informative of specific long-term sequelae like respiratory sequelae here. So we have one patient in particular called 026. So you can see in the red the result of this individual at baseline T0 and immediately after the stress test, and you can see a rapid a difference of 8-fold increase in this microRNA, which is associated with effort, so is and capacity in VO2 max capacity, but also more importantly, we discovered that this microRNA is involved in the inhibition of a repressor called Cézanne, like the French painter. And Cézanne acting as a negative regulator, so is regulating the NF-kappa B pathway, which is a very powerful inflammatory pathway. So if you remove the Cézanne of the equation by the elevation of this microRNA, you will have more lung inflammation. The good news, there are natural compounds like the gamma-tocotrienol, which is a natural form of vitamin E that can also be used eventually to increase Cézanne by a different mechanism. So again, this is purely a concept, never been tested in clinic, even though that this compound is free access in different drugstores. So, but it awaits to have the proper funding to launch a clinical trial on the right individual selected. But this test can allow us to select the right individual, and we should see in principle a repeat effect. Another microRNA, the MIA3620, again, by comparing the COL 026 to other long COVID participants and controls, this one seems to be involved in insomnia. So more elevated is the change, so between baseline and the induction of PAM, more insomnia the participant will exhibit. And this is no surprise. So the link with insomnia is reported in panel B. Panel C show you the different target of this macroRNA, and amongst of them, there's a lot involved in the regulation of hypersomnia, daytime sleepiness, and narcolepsy with cataplexy. So again, this is not random. So we think that we have something that is maybe of great interest for some specific type of long-term sequelae. Again, we had a surprise and we benefit of some patient partnership to showcase an observation that exposure of the hen in hot water for a few times, some patient, long COVID patient, reported that they lose their wrinkling of their skin. So as you see, panel A show normal wrinkling when you put the hen in warm water for at least 15 to 30 minutes. But if you do this, the long COVID patient tested here showed no wrinkling at the tip, at the fingerprint. So, and this is could be used as a good indicator of a loss of sympathetic integrity, which can be very quickly tested, although imprecise, but can be very requisite to all participants. We try to figure out what is needed to create this skin wrinkling following the immersion of the hands in the soil. And the ME-300 3620 could involve the regulation of Claudin-4, which is one of its known targets, which it could be critical to maintain the skin homeostasis and oral mucosa. So I think we are on the beginning of something here that could be helpful for some long COVID, and the test is so easy that everyone can do it at home, so we can work with the patient as a true patient partners. Interestingly, again, we decide, because due to the similarities between long COVID and ME patients, to measure some of our favorite biomarkers. And you can see that we saw some elevation of soluble SMPD in the plasma of the patient, and this further increased with the duration of their long COVID. And as well, we saw the same phenomenon with the age. So again, confirming that this effect seems to be mediated by sex hormone like estrogens, and that's why explain why more often long COVID are women than compared to men. Interestingly, you see for the panel C, D, and E using the DSQ questionnaire, different score. So if you are not producing enough, you have also an elevation of symptom severity, as you can see, with value below 15 nanogram per ml. If you are in the middle, you are in the correct or normal range. And if you have here above 23 nanogram per ml, okay, you see again your severity of your symptoms. So again, it's the same principle that I showed you with ME patient. You cleave too much, you are ending up with some more severe symptom, or if you are not producing enough, you have also some very similar severe symptoms. So this U-shape in a response is well known in physiology in many situations. So finally, I would like to summarize that I think we are fast-tracking ME research in Canada, but more importantly in collaboration with colleagues at the NIH, as well as across the United States, UK, Europe, Australia, and many places. The important is to work together to solve as quickly as possible the pathogenesis of myalgic encephalomyelitis, as well as to prevent the increase of the case following COVID-19 infections. So I show you some good examples. I hope to allow you that we can now identify the disease severity and disease progression using a panel of 11 microRNA that allowed us to stratify a patient and get more insights about the cause of key symptoms. We can predict now which treatment will be more successful, although this will await more testing and more funding to do well-planned clinical randomized clinical trials. But the biomarkers can predict the therapeutic response, and we identify new targets that are not specific for ME/CFS, but are really having a damaging effect on ME patients and long COVID patients as well. So, and we think that we can advance treatments by developing, for instance, selective blockage of TSP1 signaling, which is available and could reduce mantle brain fog as well as orthostatic intolerance and maybe POTS. And that these approaches can also should facilitate the repositioning of drugs that are used for the disease, because we know now how, where and how to target and who could benefit of such treatment. Finally, I would like to acknowledge the different funding organizations, as well as a very special thanks to all participants and families for their contribution to these studies, and as well for the long COVID studies, and to the different national organizations that assist us in the enrollment of participants. And obviously, I need a big thanks to all my team members and Dr. David Lee as a key collaborator who is based in Florida Atlantic University. So thank you all for listening, and feel free to contact me if you have any question about this presentation. Thank you.
Thank you for joining us today. My name is Therese Russo. I am a person with ME/CFS and POTS, an ME advocate, and a member of the Community Advisory Committee for the NIH ME/CFS Research Network. Today, I'm speaking with Dr. Alain Moreau, Director of the Interdisciplinary Canadian Collaborative Myalgic Encephalomyelitis, or ICanCME Research Network, and Sabrina Poirier, who is also a member of the Community Advisory Committee for the NIH ME/CFS Research Network, as well as chair of the Working Group on Training, Development, and Medical Education at ICanCME. This Q&A session is designed for the larger community to better understand the important ongoing biomedical research taking place on ME/CFS. It's worth mentioning just this to underscore that many of the viewers may not have a technical background in the sciences and may have cognitive limitations that make it all that much more important for us to deliver easily comprehensible and bite-sized information to them. Also, since our time together is somewhat limited, I might occasionally gently interject just to nudge us along to the next question so that we can try to cover as many as possible. So with that, I'd like to give you both an opportunity to introduce yourself. Dr. Moreau.
Thank you, Therese. So I'm Alain Moreau. I'm a researcher. My lab is at the St. Justine University Hospital, which is the largest mother and child hospital in Canada. And I'm also full professor at the University of Montreal at the Faculty of Dentistry and Faculty of Medicine. So, and I have the pleasure and privilege to lead the ICanCME Research Network, which is a national research network funded by the Canadian Institute of Health Research.
Excellent. And Allison, Sabrina, I'm so sorry.
So thanks so much for having me today. I'm one of the co-founders of the ICanCME Research Network, as well as a steering committee member and chair of our working group on training, development, and medical education, as you mentioned. I'm also a member of our NIH-funded ME/CFS Research Network Community Advisory Committee. I'm also a patient partner in research, a patient advocate, and a person living with ME for 12 years.
Great, thanks so much for joining us today, Sabrina. Can you talk a little more about some of the work you're doing with ICanCME, and particularly maybe what work you are most excited about?
Yes, so ICanCME Research Network is a bit different than the format that is currently supported in the United States by the NIH, having the Center of Excellence. So, and we are very pleased that the NIH recognized ICanCME as one of their Center of Excellence, and we really appreciate the opportunity to collaborate with our colleagues in the state. What we are doing is the mandate of ICanCME Research Network is to really increase research capacity in the field of myalgic encephalomyelitis and to bring new players in the field. So The network is a bit different than the Center of Excellence because we create our own internal competition with different activities and programs. So we have core grants, we have new discovery grants, we have also bursaries for graduate students or postdoctoral fellows. And we have also different activities that are not related to immediate fundings, but to bring people together. So we create several working groups. So they act as a think tank. They are also acting as our knowledge mobilization platforms, which is very important. And that allowed us to bring some answers to the federal government if there's any questions. And we also had some interaction with provincial government because health is a provincial activities in Canada. So we work with the government of Ontario, for instance, and government of Quebec for different developments. So having this recognition by the federal government and funding ICanCME research will open different doors. So having said that, in terms of scientific activities, we are covering a large spectrum of research from genetics predisposition by studying multi-generational families where you have several affected individuals. So as well as to look about DNA methylation profile where there is no mutation in the DNA, but it's the addition of methyl group on DNA that change the activity of the genes. So this is another mechanism, very important. My group personally work also on some epigenetics aspect, and we developed probably the first tool of its kind to allow the diagnosis of myalgic encephalomyelitis using a panel of 11 microRNA. So which are very small molecules of RNA that are not coding for anything, but they are interfering with the traduction from the messenger RNA in the protein. So if you have a very high level of some microRNA, you have less protein and you end up with some, let's say, sometimes more severe symptoms. And this is a very exciting new development that we have done inside ICanCME Research Network.
Wow, that's great. It's really great to hear that there's such a robust collaborative research happening in Canada. And I want to ask you a little bit more about those specific projects that you're working on right now. But first, Sabrina, thanks so much. I'm really glad that you're here today too. Would you mind talking a little more about some of the work that you are doing with ICanCME as a patient partner? And particularly the work that you are excited about?
Absolutely. So one of the things that I get to work on is I'm the chair of our Trainee Development and Medical Education Working Group. And I'm really proud of that group. We have built something really strong. We have researchers, clinicians, and patient partners all working together collaboratively and as equals. And it really makes the work so much stronger when we all work together. So one of the projects that I'm most excited about working on is the Academy, which is one of Dr. Moreau's ideas around an educational platform, something that everyone in the network can benefit from, but also hopefully eventually a larger community. And so we're really looking at how to build that right now as a team and how to create a space where we can meet the needs of different audiences and really help to educate everyone who didn't have the opportunity to learn about ME, perhaps in their training or in their experience so far as clinicians or researchers. So we're really excited about that. And another project that we have within that working group is we're working on a research project, which I can't say too much about right now, but it's a very important research project for our community. So those two projects are something I'm really excited about within our research network.
Wonderful. So Dr. Moreau, your research presentation is so robust and you have at least several exciting projects going on and it's all amazingly detailed. So you started a little into this just a few minutes ago, but our listeners who might be struggling with some cognitive dysfunction or for those without a technical background in the sciences, me included, would you mind summarizing a little more some of your primary findings for us in a few sentences?
Yes, so first of all, my team is working also with patient partners, which is very important for us in the conception of our experimental design. So, and we learned from the beginning when I started in 2015 that to really decode what is happening in neoadjuvant encephalomyelitis, we have to understand what is causing post-adjunctional malaise at the molecular level. And because this is the hallmark symptom of this condition, as opposed to any other condition where you might have fatigue or related symptoms, PAM is very unique to neoadjuvant encephalomyelitis. And to do that, we create a new device where we induce a mechanical stimulation using an inflatable cuff that we apply on the arms. And this is normally giving a therapeutic massage to healthy people, but for any patient, this is really creating a kind of exercise. And we characterize all the facets of what is happening. This is what I'm calling in my presentation, what I refer to deep phenotyping, to really understand what is happening. So before and immediately after the 90-minute stimulation, and maybe 5 days later. As far as I know, I think we are the only team in the world doing this type of longitudinal study over a week for most of our participants. And this is very advantageous because everyone agrees that PAM is highly variable between individuals and also from a same subject. It may vary over time. So we need to capture all the elements in a more objective manner, not just based on questionnaires, self-report questionnaires, but also to have more objective biomarkers or value, whatever we are registering. So that's why we start by measuring or collecting blood before, after. We are doing neurocognitive assessment using a test called brain check, before, after, and 5 days later to see if there's a recovery or partial recovery. We are measuring change in the brain oxygen level, what we call brain oximetry, and to really demonstrate, not only to the patients and clinicians, but also to my fellow colleagues in research, that the stress test that we apply through this mechanical stimulation is really inducing change that explain the development of brain fog, the development of POTS, because we are showing change that can be explained by sudden change in the heart rate, for instance, during the stress test or immediately after. So we were very delighted to work with patient partners to create this design. And from that, after that, it's more easier to look at different biomarkers. Could be circulating factor, protein, peptide, microRNA. We can further stratify patients in different categories. And the tests that we are developing, and others will also follow us doing such development, will help to diagnose ME, prognosis for ME, select eventually therapeutic approaches where we know that some drugs like ampligines or maybe other molecules shouldn't be applied because it's quite unlikely to work, as opposed to other drugs that can be very successfully applied. And this is very important for the next phase, not only for us, but for many others to design better clinical trials for ME.
That's really great. And we'll get to a little bit more about that study and particularly your inclusion of severely ill patients within that study in just a few minutes. But I also wanted to ask, you mentioned in your presentation seeing that various research data was starting to converge. What are some of the most exciting convergences that you're seeing right now?
We try to connect the dots from our own work with others people, because due to the complexity of ME, we shouldn't work in silos. Whatever the excellence of each silo, you shouldn't work in isolation, because it's impossible to make a breakthrough discovery to really understand the disease. So that's why ICanCME Research Network has been created. That's why also the NIH is supporting the Center of Excellence, that we work together. So the monthly meetings that we have and in-person meetings are game changers, where we eventually learn from each other, but also that we complement and we learn from previous results. And now we translate these new knowledge in something that can be therapeutically applicable. So the convergence, I will give you just a very specific example. The work from Ian Lipkin groups and the work with others like Maureen Anson, Bob Navio also present a lot of very exciting metabolomic results, okay, about change in lipid composition. And there are two classes of lipids that are related, ceramide and sphingomyelin. And in ME patient, ceramide seems to be accumulating, okay, up to 86% in women and around 50% in men affected by ME CFS. Nobody really pay attention about, okay, what are the mechanisms underlying, until that we discover molecules that convert ceramide to sphingomyelin. And that molecule, SMPDL3B, very complex name, okay, but it's playing a role in immunity, it's playing a role in lipid metabolism, and is normally anchored in the membrane of the cell. So the molecule must be attached. But in ME patient, there's a kind of cleavage. So the molecule is floating away, and more molecules that are away, which means they are not efficiently attached to the cell membrane, so they are not playing their role. You see the symptom, the severity of symptoms increase. So now we are trying to figure out what are the mechanisms, but also how can we find drugs that can reduce this cleavage. Rituximab is one of these drugs, okay? It's not perfect, but can be maybe very effective for some patients, but not for others, because others, their problem is not the cleavage, is they are not simply producing enough of this molecule. So if you look at the metabolomic, you will see the same situation. Very high level of ceramide, low level of sphingomyelin, but the mechanism is different. So again, all my presentation is around the needs to precision medicine for ME. Without precision medicine, if we put all ME together every time, we are increase our chance to fail to really discover what is going wrong, and to demonstrate that we can successfully reposition some drugs. So this is just a simple example about result by others, and we gave them, let's say, a different flavor, a different perspective, but that can be a game changer for the next phase.
Absolutely. I really appreciate the way that you're committed to breaking down those silos and working with others, for instance, in the field of metabolomics, and then using objective markers, or finding objective markers through exacerbation of things like PEM to get closer to biomarkers and treatments. So as you know, having a biomarker is a high priority for our ME community. We know that if doctors had a definitive test, it would help greatly with diagnosing patients, and it would go far in validating a disease that has been historically contested, at least in some circles. Do you think we're getting closer to finding a biomarker? And what are the next steps that you think there are to developing one that can be used in a clinical setting?
This is a pretty good question, because it depends what you really want to need. I think we developed a successful panel of 11 microRNAs that can be very useful, clinically speaking, diagnostic tools for physicians to really make a call at who is suffering of ME. We are submitting today a second paper about this, where we can further discriminate who is ME from who is suffering of fibromyalgia, for instance. As you know, that a lot of symptoms between both conditions are overlapping. Plus, ME also may suffer of fibromyalgia. So fibromyalgia can be a comorbidity of ME. So now this paper clearly established using this panel that we can really stratify ME plus FM versus FM when compared to each other or compared to healthy sedentary control, which is us, I think that would make some noise, like our first paper. And that would be in aid of physician and patient ultimately to understand, okay, so the condition. Now, the same tool can also identify some potential targets that we can leverage current drugs or medications that can be applied to either increase or decrease or block the activation. And what we are doing, I think for the next 5 years, you will see that we will be able to really reduce the severity of some key symptoms, POTS, brain fog, for instance, by selecting the patients based on this so-called precision medicine tools that are currently in development. And that can be a life changer for a lot of ME patients. So if you are bedbound and you now housebound, and from housebound, you can start to work part-time. Wow, this is a kind of revolutions that we all wish to achieve that. Eventually, and this is where it's the complexity, having now several biomarkers. We have an example from a patient in Calgary that we have followed over a year. I won't say the name of the medication that's been tested, but she got a full remission during 6 months, full remission. And now she relapsed and got more severe symptoms. And we saw a lot of changes in the profile associated with the relapse. So the idea is we don't want to treat the individual for just 6 months, even though that the drug seems to have some miracle effect during 6 months, because this is not a long-term solution. So we need to build and say, okay, what we need to remove or to add, and what are, let's say, the most important marker for that individual that will help us to further define the best therapeutic treatment for that individual, which won't work for the next one. So again, I'm stressing my point about the necessity to embark the whole field in precision medicine. And even in my lab, we are never comparing just ME against LT control or against fibromyalgia, because a lot of things are disappearing. Just separating male and females, you can start to see a very different profile. And as a patient, you already know that you are not reacting the same way than a patient, a man suffering of ME. And clinically speaking, we need to understand that and translate that to better molecular tools and better treatment. So precision medicine should be our leitmotif, and we need to keep that on track.
Oh, great. I love the way that it sounds like you're centering precision medicine to kind of better understand all of these different subgroups, given that ME is such a heterogeneous kind of patient population, not to mention the different demographic and other factors that we also have to take into consideration. So that's incredibly exciting to be thinking about refining treatments based on that. So this next question is for Sabrina first. So earlier, Dr. Moreau mentioned the longitudinal study that they're doing where they induce post-exertional malaise, then do deep phenotyping on patients as a way of continuing to find objective markers of disease. And in this study, there is a cohort of severely ill patients. So to remind our audience, the severely ill are those who are housebound or bedbound, and they account for about 25% of all MECFS patients. So we were really happy to hear that they're being included in this research study since they're often left out. And Sabrina, as a patient, I'm just wondering if you'd like to speak a little bit about to the importance of including this particular patient group.
Yeah, thank you. I think so often in research, I think this particular population has felt left out, and we haven't been able to collect enough data. We haven't been able to really explore what their feeling of the illness is, what their experience of the illness is. And I think it's really important as we move research forward to really start focusing in on this group and finding ways to build research projects that are inclusive of a wider demographic. In many ways, severely ill being one of those groups that I think have been missed, but also our BIPOC community are really important as well. And we know we need to do better there. So I think it's just really exciting to see a researcher focusing on that and building a research project to understand how to be able to meet them where they're at. And so in Dr. Moreau's research, they're actually going to their homes, they're taking samples. And I think that's really somewhat unique in our community and something to be celebrated and get excited about. And I look forward to seeing more researchers wanting to study this group. I myself am in this group. And so it's hard sometimes to be able to get out and participate in research if it's one, not in your area, or two, if you're too ill or it takes too much from you to participate. Then you really have to constantly be weighing that. And I think this is a really exciting way to be able to include them. So I'm really happy to see this. I really hope we inspire other groups of researchers to start to think about how to build research projects inclusive of this community. And I think working with patient partners from this level of severity could be a great way to understand how to do that even better.
Excellent. Dr. Moreau, can you share a bit more about how you were able to include severely ill patients and why you felt it was important for the research to do so?
I would say that all the points covered by Sabrina make a lot of sense to us. And this is what we had at the inception of this experimental design, where we decide from the inception of the project to really address this issue on at least housebound patient. If we have to make a breakthrough and eventually change and have an impact on their life, we have to work on moderately to severely affected individual. And I'm not saying that the patients that might have mild symptoms are not severely affected too. Those symptoms, as you know better than me, are fluctuating over time. And because it's a silent disease, it doesn't appear on your face. So that's why we are sending our clinical research nurses at home with all the gear, with all portable machines to do that. And I'm very happy to report that this inspires other teams. So for instance, Professor Jonas Bergquist are doing at home now, doing collect of samples on housebound individual in Sweden. He's not exactly using the same technique and same approach, but he got some inspiration. We have recently a discussion amongst our steering committee meeting about to replicate the protocol I'm using in Montreal to transfer the knowledge and protocol in Toronto, as well as the other clinical side for MECFS in Canada, where they are in Vancouver and Halifax, to allow the different teams to not only learn, but also further develop the test. And there's a lot of excellent clinicians where they need better tools now that, and I'm sure again, that by allowing and supporting replication studies independently, will further confirm the clinical utility of the protocol, will improve the protocol, okay? And I don't think the inventor should be left alone. And in fact, we should work with others that will bring to a new level the test and how can we use and interpret that. Because at the end of the day, we are developing the tools to improve the life of Canadians and everyone in the world suffering of ME/CFS, as well as to how can we further assist physicians. We have a lot of physicians that do not have any expertise, any training in ME/CFS, and this is hurting us big time. So by providing tools, and I will give you just a very nice example, a very well-known physician in Montreal, I won't say the name, contact me suddenly when our first paper is being published in scientific reports and say, you don't know me, I'm working in neuroimaging at very nice institution. I always refuse patients. But since I saw your paper, now I believe this is a true disease. And now I will start to accept and test the patient using all my capacity, my expertise, and our world-class platform to neuroimaging the brain and what is happening in those patients. So that happens almost overnight following the publication. So this is my point when I have discussion with my colleagues is we can art, we can claim things, but when we publish outstanding science and this science is read by others, we can change the field. We can change the medical fields by bringing best research about ME/CFS. This is the only way that we can turn around the table about that disease and to bring other people with great expertise now to pay attention. So it's not just to be recognized by NIH, by the CHR in Canada, but it's also to let's publish the best knowledge, improve the knowledge, and bring these studies that can be game changers. So this is a just sample example that someone that I have no idea who that individual was, and now he's a collaborator, and now he's accepting as part of his medical mission, ME patient, although he refused them during the last maybe 20 years. So this is a very simple, direct example that one publication can be a game changer and bring new players in the field.
Absolutely, yeah, and it really highlights your role, you and your team's role, and Sabrina and her team's role in kind of almost a mission-driven kind of work to continue to educate clinicians and move the field forward in understanding a variety of aspects of this and similar diseases. So thank you so much for all of that work. We're already running short on time, but I wanted to ask you two more questions. We were really pleased also to see that you use sedentary controls in some of your studies. Why is it important to the research for controls to be activity matched? And can you review how this helps distinguish ME/CFS from healthy people who are deconditioned?
This is a pretty good question again, because in the real world, you have people, especially adults that are aging, they have different comorbidities. They may have cancer or bladder cancer in the past. They have osteoarthritis, osteoporosis. So this is the reality. So if you are too keen to select perfectly healthy individual and you have very specific subgroup of ME, you may end up that your marker A is so different and they are never matching. The marker on my hands. Here, if you use if you select real life situations, so what I mean by pragmatic tests, you start to see that the background of the noise is going that, and now you are losing. So your markers are not clinically useful because they are masked by other comorbidities that you did not expect. Things might happen about change in your sexual hormones. So when a woman's reach menopause, or whether they receive hormone replacement therapy or whatever the drugs, we have patients receiving 12 different drugs for different symptoms. So you cannot just clean this and say, okay, don't take any drug during a week and we will test you and see what is happening. So we are taking people as like taking a Polaroid shot and see what's happening. So that's why the stress test, so the before and after, we neutralize these effects. So, and especially because a short time of the stress test, 90 minutes, allow that if you are taking 9 different pills, and we start to test you in 90 minutes later, the 90 minutes cannot erase the different pills suddenly. So we are integrating these results. Same if you have OA or osteoporosis or other conditions. So that's, I think, bring an added values of what we are doing, because in the real world, we have to deal with that. It's not a paper, it's not. So we have to move away from the academic field where I'm selecting so well my different subgroups that, of course, at the end of the exercise, I will be right. I will have boom. And when people try to replicate the result with a different cohort or the different setting or different drug, and think, poof, everything disappears. And this is just another useless paper in the field. Good for the researcher, good for the CV, maybe good for the ego, but is nothing good for the patient. I want to stay away from this situation. So we are challenging ourselves a bit higher, but that's the only way that we can decode MECFS.
Excellent. So last question I wanted to ask about treatments, because of course that's something that the ME community is always very interested in. Do you see any potential for treatments or interventional studies in the future resulting from the ongoing research you've been doing? And you know, when you're answering this, feel free to speak to the reevaluation of the rituximab trial data that I understand you've been working with, as well as any other promising drugs your research suggests should be tested with the idea of repurposing them for MECFS.
So again, this is the several $1 million question. Okay, what can we do? Or what can be done on ME patients? So it's one thing say that, yes, we know that you are sick or sicker, you are in that subgroup, yeah, but what can be done? So again, keywords is precision medicine. You raised the issue about the rituximab clinical trial. We had a nice collaboration with Dr. Ongstein from Norway and his colleagues, Professor Miller, where we blindly reassess different biomarkers in their plasma. So that's why the condition that we test sample blindly, so we didn't know who's controlled, who received the albumin or received rituximab. And when we agree, we share the result with them, they look by themselves. And that replicate one of the markers that we mentioned today, SMPDL3B. So soluble version of this is associated again with Norway's samples in the severity of the sample based on their own criteria and based on their own analysis. So this is not published yet. That will be part of the pipeline of publication that will be submitted soon. But this is important for us, independent replication, different population, and we end up with the same conclusion. Now, whether or not rituximab is working, now it's a question about how you will select patient, because I told you that ME are not equal, they are different. So we raise some issues about if we select only patient with high level of soluble SMPDL3B, and now whether or not rituximab is better than albumin in terms of improving the symptom, and we got the confirmation in their own study. Maybe 2/3 of the patients wouldn't have been eligible to receive rituximab because they are not presenting this selection criteria, having high soluble SMPDL3B. But for those, we saw some very strong glimpse of hope that it seems to work. Okay, not perfectly, but seems to work and make sense. Same with the albumin. Albumin is not neutral here, has been also raised as an issue in many other previous studies by others. So we need to pay more attention about some details. I don't want to go here because it's too technical, but there are issues using albumin as a placebo, not due to albumin, but to the whole preparation of this the solution. But 5 years ago, I would have said, oh yeah, we have maybe rituximab, maybe few other drugs. Now I can, when I have presentation with my colleagues, I can fill a table with a lot of molecules based on different biomarkers. So now the option based on our own work is maybe up to 25 different drugs. Sometimes the drugs will increase some specific biomarkers that need to be increased. And I would need to take a drug that would do the opposite because now the biomarker is too high for this individual. So if the field think that there will be a magic bullet to treat all the patients the same way, I don't think that will happen. Not because it's a MECFS, because all complex chronic diseases have never been successfully treated by a single way. So that's why we need to not only personalize the treatment, but to be very precise. And maybe one drug can do wonders during few years, and eventually we need to change the treatment for a patient if there's a kind of relapse eventually. So that's why we are at the beginning of this new era of treating patients by repositioning drugs. But now I think for the very first time, we have tools to select the patients. And that will reduce that will reduce the failure of these different clinical trials.
Great. Well, Sabrina, I just asked Dr. Moreau to end on talking about maybe some promising treatments that could be in our future. I just wanted to check in one more time with you about whether there's anything that you're excited about in the next 6 months or year of your work with ICanCME that we should be on the lookout for.
Yeah, so I think the Academy definitely building that educational platform and really being able to bring different audiences together to learn about ME. We've seen a real surge in interest since long COVID has become more discussed, more understood. We're definitely having more clinicians, more researchers reaching out and asking us, you know, where do I go for good quality information? What's the science on this? And so we've been pulling that together sort of ad hoc every time we get a request. And the exciting part will be finally having a place where the materials are well vetted, well sourced, safe for patients, and being able to create that place on the internet in time where people can go to on their own anytime that they want to learn what's the most exciting and up-to-date information. I think that's a really important foundational piece to Canada's moving the needle for ME patients. I think it's something that's been missing, and so I'm really excited to help build that. And I think also some of the research projects that are coming out of the network have a lot of potential to really help us understand another perspective from clinicians on what's working and what isn't, where the gaps in knowledge are, and so that we can better understand how to help them do their job as effectively as possible. Because I think a lot of what we've learned, I think over the last few years, is that a lot of clinicians had never heard about it or had heard incorrect and outdated theories. And so when we give them correct information, we really do see a shift in them. And I sometimes hear from them saying, you know, I tried this with a new patient and it worked. And I think understanding comorbidities is a big part of this as well, really being able to look for those comorbidities and treat them so that patients' quality of life can be improved now while we wait for those treatments that Dr. Moreau and others in the community are working so hard on. So that's the stuff that really excites me as a patient partner in our network. And I think I feel really hopeful about where science is headed.
Excellent. Well, thank you so much, Sabrina, for joining us and helping us to understand more about the work you do. And Dr. Moreau, thank you so much for taking time out of your busy schedule to give us an overview of this research. I just am so grateful to both of you for the impact that you're having on the field. And I just can't wait to see where your research continues to go. And I hope to hear from you all both again soon about additional progress. So thank you so much. And with that, I think that we can sign off.
Thank you, Therese.
Thank you.